Thank you for asking me to talk about my laboratory's work on the HIF pathway, which of course is the target of the new treatment for anemia that we're discussing today. This was my question as a young nephrologist 30 odd years ago, how to link the regulation of erythropoietin to what we believed would be a rather specific oxygen sensor in a specific type of cell in the kidney, of course, making erythropoietin. Now, there was a new opportunity for these studies since the erythropoietin gene had been identified. So our aim was to move from the identified gene sequence through what we thought would be regulatory sequences at the gene locus towards this oxygen sensor. So our first task there was to find a cellular model in which that type of molecular approach could be applied. And this was one of the early things we did. We tried to grow the EPO-producing cells out of kidneys by expressing, as you see here, T antigen, that's an oncogene, within the EPO gene sequences with the aim of making these cells grow out of the kidney into culture where we could study them. Now that experiment failed. We never did establish those renal EPO-producing cells in culture, but did identify the cells, as you see there outside the tubule, as the rather unspecialized kidney fibroblasts that we now know do make EPO. So although that experimental approach did not deliver what we wanted, it did suggest that the cells and the property might not be so specialized. The field was, however, opened by Franklin Bunn, who derived that cellular model, not from the kidney, but from hepatoma cells from the liver, which produce EPO, as you see here, when he reduced the oxygen tension in culture. And it was Frank's cells that provided my own group and others with the substrate for doing this type of experiment, which was to chop up the erythropoietin gene in order to isolate control sequences, which we believe would lie at that locus, and identify them as the first stage in moving outwards to the oxygen sensor. So here they are. This is the black bit that we identified lying at the 3' end of, in this case, the mouse EPO gene. When coupled onto a reporter gene, we can see the effect. Adding those sequences to alpha globin made it inducible by hypoxia. So that was rather clear and neat, perhaps unsurprising, because those cells must have a regulatory sequence. It provided the substrate, however, for what was perhaps the simplest and most important experiment we ever did. What would happen if those sequences, which must interact with the oxygen sensor, were expressed in other cells where it was not believed there was going to be an oxygen sensor? And this is the result. We found practically the same behavior in all sorts of cells, which had nothing to do with making EPO, not from kidney. And the implication, of course, was quite significant, that this was not a private oxygen sensing system, but a general one in cells that are not making erythropoietin. So it must clearly be doing other things, and that opened a very wide field. The proteins binding that sequence were identified by Greg Semenza and called HIF, hypoxia-inducible factor. The implication, of course, now was that HIF was in all cells and doing other things. We found the first genes that respond to HIF, other than EPO, as the phosphoglycerate kinase gene. That's a glycolytic gene, and indeed all glycolytic enzymes are regulated by this system, perhaps slightly surprising given the very different dynamics of glycolytic regulation versus EPO. This is exemplified as part of our work in the red. I've circled the HIF, which is a protein retarding this labeled oligonucleotide in a gel. So where you see these black bands up here, that's HIF binding to the response element. And you see this experiment was conducted in insect cells, and it implied that the insect cell, the insect, of course, not having EPO, not having a blood vascular system, still had the same system of oxygen sensing through HIF, which of course must in the insect be working on other genes. But at the time, I don't think any of us had any idea of the panorama of what was to unfold. Literally thousands of transcriptional targets of HIF involved in many, many processes, both directly and indirectly interfacing with the delivery of oxygen homeostasis and oxygen balance. I'm not going to talk much about that work at the moment, since our quest at the time was to move again upstream from HIF, which Greg Semenza now identified the cDNAs, the encoding genes, enabling a rather similar approach to be taken to finding the next steps upstream of HIF. So again, this is what we did. We pasted bits of HIF gene onto reporter proteins, fusion proteins, to try to identify which bits of the HIF gene could confer oxygen sensitivity and therefore must be interacting with mechanisms upstream. And we found three regions, rather surprisingly and rather gratifyingly, because we expected within the three regions, which must interface with the oxygen sensing signal, by which time we knew that this was also responsive to ion chelation and in line with classical observations on cobalt-induced erythrocytosis with cobalt. So we believe these were part of the sensing system. They generated a signal, must interact with each of the three areas. So we believe there must be something in common in those areas that would lead us to the next stage. Sadly, we couldn't see much in common. And indeed, in this region, Chris Pugh, who worked with me throughout this time, mutated every phosphoacceptor, since it was believed that this would be a protein phosphorylation cascade, made no difference to the regulation of HIF. And that was a rather surprising finding that diverted our attention to the possibility of other types of signaling mechanism, which indeed turned out to be the case. But we were stuck at that time. It clearly wasn't protein phosphorylation. We couldn't see a lead for the next step. That, in fact, was provided by human genetics and the genetics of kidney cancer again, which is commonly defective in the von Hippel-Lindau tumor suppressor. That familial cancer syndrome, von Hippel-Lindau disease, being associated with excessive blood vessel growth, some of these tumors associated with excessive erythropoiesis, giving a clue that there might be something about the VHL gene that was relevant to this oxygen sensing system. And indeed, that had been suggested by others, Rick Klausner, Bill Colin, Mark Goldberg, but they didn't immediately understand the mechanism. They could see that some of the transcripts that we described as being hypoxic responsive were indeed upregulated. But we were able to define the mechanism, the connection to HIF. And for the junior people in the field, it's important to realize the reason we were able to do this was that we had immediately made antibodies to the HIF proteins, including two isoforms, HIF1 alpha and HIF2 alpha. If you want to lead a field, it is very important to have your own reagents, and I strongly recommend that you devote significant capacity in your lab to making antibodies when you need them, rather than waiting for them to appear in the reagents books, when of course they're going to be available to all your competitors. So we were able here to show with great clarity that in VHL defective cells, HIF protein there was not responsive to oxygen, whereas when we put VHL back into those cells, we restored the oxygen sensitivity. And there you can see the highly dynamic response, by which time we knew was due to proteolysis of HIF, destruction of HIF in the presence of oxygen. Now, VHL had been suggested to be a ubiquitin ligase that tags protein, in this case HIF, for destruction. In this case, it was a conditional ligase. It only worked in the presence of oxygen. So that was a model. In oxygen, we destroy HIF. Without oxygen, HIF escapes destruction, builds up, and regulates all those responses that I showed you earlier. So the question is, what was regulating that interaction? And this is a classical piece of biochemistry. The treatment with a cell extract of HIF peptide did something that enabled that binding. The question was, what is it? And we conducted a wide range of mutational biochemistry, mass spectrometry experiments on the HIF peptide. And this was the eureka moment. We eventually deduced that the modification might be hydroxylation of proline. And here the evidence that it is, that we synthesized a hydroxyproline containing HIF peptide, which now no longer needed treatment with extract to bind to VHL. Why was that a eureka moment? Because the known prolyl hydroxylases, collagen prolyl hydroxylases, which we were able to exclude as not responsible, yet those enzymes are oxygen splitting enzymes. They are inhibited by iron chelators and cobalt. So they had very good properties for this action, although clearly this wasn't the collagen hydroxylase, and we excluded that biochemically. But we were looking clearly for an enzyme of the same type that catalyzed hydroxylation of proline in HIF, promoted proteolysis. So what was that enzyme? This was now another piece of biochemistry. I want to take you just slowly through the stages here, which illustrate how knowledge builds on knowledge in quite unexpected ways. My great colleague, Chris Schofield, who I hadn't met at the time, was working elsewhere in Oxford on antibiotic synthesis enzymes are related to the 2-oxyglutarate dependent oxygenase family, of which the collagen hydroxylases were members. So this is the diagram of the structures that Chris obtained, which enabled him to predict the existence of 60 or 70 of these enzymes encoded by the human genome. A quite different line of work had previously been pursued by Bob Horowitz, painstakingly catalyzing worms with defective body form, in this case 145 worms that couldn't lay eggs due to a neuromuscular defect. And here are the eggs within one of Bob's worms. We had, I'd say we were interested in primitive organisms. We had identified a HIF orthologging in C. elegans in those nematode worms. Again, we made an antibody which proved critical in the way forward. And here we see the worms inducing their HIF when simply placed in a bell jar with low oxygen. Turned out that one of Chris's predicted oxygenases corresponded to one of Bob's mutants. So the pre-existence of a mutant enabled us to test the candidacy of that as the oxygen sensing prolyl hydroxylase. And this was another eureka moment. I remember Andy Epstein, who did this work, bursting through the office door saying, "Eggle9, here's your gene." Eggle9 was one of Bob's egg-laying defective worms and was a candidate suggested by Chris Schofield. As you see, the mutant worms stabilize their HIF irrespective of oxygen. And that's the very simple system then that we discovered. a regulatory oxygenase places an oxygen atom on the proline ring to create an alcohol group. That alcohol group hydrogen bonds to the destroyer VHL, which transduces oxygen-dependent proteolysis. Looking at the entire animal kingdom, this system is present in all animals, but as far as we can see in its assembled form only in animals. With vertebrate evolution come gene duplication events, so we now have more than one HIF and one of more prolyl hydroxylase, which are now the subject of the anemia treatment that we're going to discuss. So this is the system that we found out by intention. This is what we found out in parallel, unanticipated, and what was also unanticipated at the start of the work was that these enzymes would be eminently druggable by these two oxyglutarate co-substrate analogues, which block up the catalytic site to make the cell think that it's hypoxic in the presence of oxygen. And that's what several companies have developed as HIF prolyl hydroxylases for the treatment of kidney anemia. Of course, what we have is the uncertainty, which you will need to discuss in future sessions, as to whether this pleiotropic effect enhances erythropoiesis in a way that's productive and better than giving erythropoietin alone, or whether some of these effects might be beneficial on other systems, or indeed entrain some risks as we go forward with clinical treatment. So I'll stop there. Thanks to a lot of people who've worked with me over the years. This is my group on a nice day in Oxford. Particularly grateful to the funders, the Wellcome Trust, and now the Ludwig Institute of Cancer Research, and to these three people, Chris Pugh, Patrick Maxwell, and Christopher Schofield, who are involved with so much of the work. Thank you.